Okay, I will. So this is going to be answer key video for second exam, and we won't have any assignment associated with this exam. After the first exam, we had an assignment to, correct some answers. We're not going to do that with this exam. We did it with the first exam because people were, still, learning about what to learn and how to learn about it, the topics in this course. So very much like the first exam, we have some fill in the blanks and so forth like that. And so what is one of the gap genes in fruit flies? So remember we had a handout that included, image of the female reproductive tract in fruit flies, but also had about a dozen genes down. And so one of them was a gap gene. And the gap gene that we learned about from that handout was hunchback. So it's in a group of genes that gets turned on by BIO305 transcription factor and other maternal factors, and the gets turned on, in the embryo after that first couple of hours of rapid nuclear division. In the biostula of a sea urchin, the cells initially are all holding on to this hyaluronic acid. So we wanted to learn the actual names of a few of the components in the extracellular matrix, just so we can have a feeling for what that is made of. And it's made up of lots of proteins and lots of polysaccharides, some of which are like hyaluronic acid. It's a polysaccharide, polymers of a complicated sugar. There are many, many different kinds of sugar, and they can be chained together to make polymers, and that means they have a lot of information. The extracellular matrix is structural, lots of polymers, but also lots of different kinds of molecules at different concentrations with different modifications and so forth. What protein blocks DPP? And so that's that short gastrulation, gets activated by that dorsal transcription factor binding to the regulatory region of short gastrulation in the eventual ventral region of the dorsal of the fruit fly, and working as a secreted signaling factor to physically bind and block DPP action. What are the gap junctions between egg and nurse cells called in fruit flies? They're called the ring canal. And so, these are some questions that came from the individual stories that we told about chapter 2. And so you need to understand how the ring canal fits into the story, but also to be able to spot it when you have just a fill in the blank question. What does not form in a plant that has a no mutation of leafy? So leafy is a stem cell identity gene that is activated when the vegetative growth stops and reproductive growth starts. And so leafy is one of the genes that gets activated by the FT transcription factor when the FT RNA that was made in the leaf gets translated into FT protein transcription factor in the apical meristem and allows that FT to bind to genes like leafy and activate them. So if we had a null mutation, so the leafy was inactivated, what we would not get is we would not get flowers. So kind of a funny name, because what the mutant phenotype looks like is it's just lots of leaves. It's very leafy. What is the term for a genetic factor that cannot be rescued by the fathers? And it's a maternal factor. And that's that. What family of transcription factors are expressed in the leaf primordia? And we just inserted that because our book talks about a lot of individual genes, but the Knox family of transcription factors that are stem cell identity genes in plants and have orthologs in many, many plants is a good word for us to remember and think about with regard to plants for those reasons. What part of Arabidopsis is day-length detected? The answer to that is detected in the leaves, but we got to get the signal to the apical meristem, the shoot meristem, for it to transition from vegetative reproduction to flowering reproduction. Where is collagen protein located? So you want to say the extracellular matrix. Probably everybody knew before they started this class that the collagen is part of cartilage. Well, cartilage is a form of extracellular matrix, and there's lots of collagen in the extracellular matrix beyond just cartilage, although that's a great first thing to learn about extracellular matrix is its structural role. Which meristem makes tree rings? And that one is the lateral meristem. In a trunk, it's called in a tree trunk, it's called the cork cambium or the cambium. What is the name of one Hox gene? The one we learned of, again, off our handout. And again, that was handed out because we wanted to focus on some genes that would be able to for sure identify like this. Antennapedia. Not many students got this correct. Almost everybody wrote answers of other ones that were familiar, other genes or products were familiar, with exotic hedgehog. But Hox genes are a gene family that are involved in anterior posterior patterning in all vertebrates, all animals that have an anterior posterior axis. So this one was one that was maybe largely from the book from chapter 8, but was mentioned in class in response to a question. So muscle cells require MIF5. MIF5 behaves just very much like MyoD in its role in muscle differentiation. In fruit fly ovaries, what signaling factor turns off expression of pipe? And the answer is gherkin, secreted from the egg and affecting the surrounding follicle cells or the nearby follicle cells. What is an early fruit fly embryo called when it's just one cell containing hundreds of nuclei? So that when that happens in an animal, that's called a syncytium. So a syncytium is a bag of nuclei, and so the specific term for this embryo is called a syncytial blastoderm. What's a hormone that causes apical dominance in plants? And that's auxin. Okay, like that. Okay, so on to these. Briefly describe one example of homology in evolution and one example of analogy in evolution. And I think everybody got this right. All but one person used the example from class. That's an example that you probably learned of before in school, and that's the idea of a bat wing and a dog leg, or any two vertebrate forelegs being homologous, common ancestry, and then a bat wing and maybe a dragonfly wing as being analogous, not having a common ancestral origin. One or maybe two students used examples that weren't ones that were based on, kind of these physiological examples, but rather talked about ones relevant for our course, like, for example, the analogy of stem cells in animals and plants, and the homology of the stem cell identity factors in animals, for example, but not the same ones in plants. Draw and label a neural crest cell migrating, and students did overall a really good job, really adding a lot of details to this. And I hope to get that with this, hint of making more than one drawing. And just a couple things, where people left out, they didn't write about the, chemotaxis, or they didn't write about the actin microfilaments. They just wrote about the microtubules. So, students had a lot of detail, so that meant I expected a lot of detail in this answer. And in general, people did a great job on it. Great. What are two types of cells in fruit fly ovaries that help with egg maturation? And so a handful of students put the stem cell, the germline stem cell, and that's true. They don't really help with egg maturation. They're used to make the egg initially, and but I could understand that answer, and kind of gave them partial credit for going down that pathway. And then I think the one example that everybody gave was a nurse cell, and the other one that I was looking for that most people gave was the follicle cells. And then you had to give specific examples, describe one role that each cell type plays. And so we learned about the nurse cells making bicoid, for example, and so you could write that out in some detail. And then follicle cells, well, we learned about them making pipe, and so you could write about that in some detail. And but that's what we mean by an example, is a pretty specific example that we learned about. similarities and differences in plants and animals. So in general, people did a great job with this. The examples are about development, and not just, oh, plants photosynthesize and animals don't. It's a very different focus here, right? It's a focus of our course. And I won't talk through the list of the examples, but in general, people did a good job and recognized that don't just list them, describe them, right? So, paying attention to that language, right? That's different than when you say list something. It just makes the list. You don't have to explain them, but you really want to work this out some, okay? And but yeah, again, I won't talk through it. If you have questions about it, I think just let me know and we can talk through it. But in general, people had solid answers, and people who lost points just lost generally a couple, except for if they were using examples that weren't about development, that really weren't part of our course. Neural cells are only found in vertebrates, and they're in all vertebrates, and we learned that they play a role in the making of the face and the pigments of our skin, epidermis, in all vertebrates, and also the peripheral nervous system, the sense of touch and kinesthesia. They actually play a few more roles, relatively minor to these, and these are just really memorable because they're such a huge part of the story of vertebrate bodies. Segment, compartment, segment boundaries in fruit flies. So that's that macaroni assignment, and that we did, where the macaroni noodles were examples of cell adhesion molecules. And people did a really solid job with this first part. The second part, it was surprising to me or interesting to me that students usually did a good job listing 5 genes expressed in the posterior compartment, but maybe lost points on trying to describe what they were, because we made a list and we had specific reasons for each one to be on that list, and you didn't have to give the exact reason that we created, but it had to be a solid explanation of understanding not just a list of genes, but why those are part of being a posterior compartment cell. Because when we say that a cell is a posterior cell, it's because primarily, ultimately, fundamentally, it's expressing a set of genes, and those include genes that are specifically making the cell be posterior. Name 3 major types of cell adhesion molecules, and so we had the cadherins, the CAMs, and the integrins. And students did a great job on listing the names, and an okay job, overall for the class in describing an important feature of each. And so cadherin, yeah, it's calcium. It also is, to describe it, it's calcium dependent, and it's binding cadherin molecules on 2 different cells. Also, it connects the 2 cells, the cells cytoskeleton to each other, so it just plays a structural role. Also, that's where beta-catenin is, and beta-catenin gets released when the calcium levels drop and the cadherins let go of each other. So as a part of that, students were losing points pretty often because they didn't talk about the signaling role. So an important feature of each of these is that they play signaling roles, not just structural roles. And you can see that one was worth 10 points because there had to be a pretty solid answer. These are new to us, but we really kind of spent time explaining them. Then the last question is define radial intercalation, medial intercalation. And radial is when you have layers of cells that flatten to a single layer, and as such they spread out, not in a cell migration way, but just in a kind of jostling way. And the example of that in frogs is when the ectoderm spreads during gastrulation to cover the surface of the embryo. It's initially just up here in the animal pole, and it needs to spread, and does that by radial intercalation. Then medial-lateral intercalation is when cells are all on the same level, but they rearrange to make a different shape. And in our case, the belt of mesoderm is going to undergo medial-lateral intercalation. It's going to get narrower by having the cells slide past each other, And in getting narrower, the belt needs to get longer. So medial-lateral intercalation explains that. Now that's it for our second exam. And if you had questions that you hoped were on the second exam that weren't, you should email me those questions to suggest them to me, because I'll put them on the final. Because there are questions on this exam that got left to be put on the final. And so we're going to do that in just a couple of weeks. And so again, check it out, and if there's stuff that you really wished it could have been on there because you spent time learning it, let me know. And then also, if you have questions about the exam or the corrections, answers to the exam, let me know about that. Let me know if I added your scores up wrong, and we'll follow up with this and make sure that we head forward with some confidence. You want to go through this exam to make sure you understand anything you got wrong, because again, our final's 2 weeks away, and I'll ask different exams, but also different questions, but also some that overlap with the first and second exam, to make sure, kind of confirm that you actually went back to those exams and, put more energy into learning the material even after the exam. Great. Okay, so that's it for now.